Hi everybody, today we're going to talk about Gelelectrophoresis, and to understand Gelelectrophoresis we need to understand the parts that make up the name. Phoresis, it means migration or movement. Electro, we will use electricity to make the DNA migrate, and gel, we will run a current through a gel containing our DNA samples. Gelelectrophoresis is used to separate DNA fragments according to size and to determine the length of an unknown DNA fragment. But first we need setting things up, and for that we need a chamber. This chamber will hold our gel. We need a power supply which will deliver the electrical current to the chamber and the gel, and we need wires to connect the power supply with our chamber. But it's gel electrophoresis, so we also need a gel. And for a gel we are going to use Agarose gel. This is a gel made with Agarose, which is a polymer derived from seaweed. When mixed with water, the Agarose polymers will cross-link and they will form a molecular maze. The newly formed Agarose gel will be poured into the chamber, and then we are almost ready to perform our gel electrophoresis. The only thing we need is wells. We're going to prepare our wells by placing a comb inside the newly poured gel when it's still wet. When the gel hardens, the comb is removed and the teeth of the comb will have formed wells in the gel, so we can load our DNA samples. But before we're going to load our samples, we will fill the outside wells with a DNA ladder. This DNA ladder is a mixture of DNA fragments with well-known lengths, and we're going to use these lengths to estimate the length of our unknown DNA fragment. And our next step is loading our DNA samples. These DNA samples consist of our DNA fragments with an unknown length and a dye. This dye is very important to monitor the migration of the DNA through the gel, because DNA is invisible for the naked eye. So we're going to mix our DNA with a dye, and then we are going to load our DNA samples into the gel. We're going to load our DNA samples at a negative electrode of our gel. In this case it is the left side, the side with our black wire. When that's done, then we can start with our DNA migration, and this is done by adding an electric current through our gel. The negative pole is connected to the DNA sample side or the well cell side, and the positive pole is connected to the other side. We will add a current. The DNA fragments will migrate through the gel to the positive pole. Only the dye is visible under normal light. The electrical current is switched off when the DNA dye reaches the edge of the gel. But to understand why the DNA will migrate through the gel, we need to zoom in on the chemical structure of our DNA fragments. DNA consists of nucleotides, and one of those nucleotides is formed by an organic base and a sugar phosphate backbone. And to find out our answer, we must zoom in a little bit further on the sugar phosphate backbone. The DNA phosphate backbone consists of phosphate, which contains a lot of negative charges. The DNA fragments are attracted to the positive and repelled by our negative electrode because of these negative charges. So our DNA fragments are attracted by the positive pole, our longer fragments will migrate slower, and our short fragments will migrate faster. This means that at the end of our gel electrophoresis, we will find the shorter fragments of DNA near the positive pole, and we will find the longer fragments of DNA near the negative pole. For our last step, we need to visualize our DNA fragments. We're going to do that by adding a positive charged DNA dye that will bind our DNA negative charged DNA backbone. When we turn on a UV lamp, we will see that our DNA will become visible. Thanks to our DNA ladder that's on the edges of our gel, we can estimate the length of our unknown DNA fragments. And that's it. That is DNA gel electrophoresis. Now you know what it's all about. I hope you really enjoyed this video. Give a thumbs up if you liked it. Thanks a lot for watching, and see you next time. Bye!